Hey everyone, and welcome to Chapter 44. In Chapter 44, we are going to pretty much exclusively be thinking about homeostasis in the context of salts and waters. How do we balance how much salts we have? How much do we balance how much water we have? How do we do all that fun little interplay? This chapter has always kind of held a little bit of a special place in my heart. For the kind of intro photo on this one, I choose to always use my former dog. His name was Winston. He was in some ways a good boy, in many other ways not a very good boy. But kind of a thing about him that kind of characterized his life was he had kidney disease, and that is ultimately what caused him to pass away back in the summer of 25. But he was a trooper throughout all of it. And as I got to learn more about the urinary system while I was in grad school, while I had him, the more I got to appreciate just how crappy his kidneys really were and why he constantly liked to pee on things, sometimes when it was appropriate, many other times when it was inappropriate, often holding his favorite toy all throughout the process. It made sure I kind of contextualized and didn't blame him for when he wanted to pee in the house because, quite frankly, he couldn't osmoregulate as well as he should have been able to because of crappy genetics. He was a good boy, though, so I always like to kind of think about him as I go through this chapter. So, kind of transitioning from that, though, let's get into it and think about how and why we actually need to maintain proper solute balances. This idea of osmoregulation. Osmoregulation is just going to be the term for the general process of controlling solute concentrations. That word osmo should seem somewhat familiar to you because we have talked previously about water moving through osmosis, something down its relative gradient. It's another chance for me to get to draw this exact same figure. If something has a high gradient, it wants to move to an area with a lower gradient. In this case, it's the relative movement of water down its gradient, typically through some selectively permeable membrane. And so when we use the term osmolarity, we are referring to the measurement of how solute something is, how many solutes are in that solution. The more salts, something like sodium, something like chlorine, the higher the osmolarity. Pure water would have a very low osmolarity. Seawater would have a very high osmolarity. What you'll typically see in a lot of these is the measurement of milliosmoles per liter. Since this is a measurement of something in a fluid, you always have to give it context for how much fluid is there. And typically, milliosmoles is the one that is most appropriate for a lot of the things we're going to look at. For context, human blood is somewhere around 300 milliosmoles. Oops, I forgot my little S there. Milliosmoles per liter. Whereas something like seawater is usually around 1,000 milliosmoles per liter. Just to give you kind of an idea of a range of how salty something is, if you want to think about it that way. And then to kind of revisit a couple of terms that should be familiar to you, except instead of saying one suffix, we're going to give it a slightly different suffix, are the terms isoosmotic, hyperosmotic, and hypoosmotic. You would remember these probably by the terms isotonic, hypotonic, and hypertonic. It is basically the exact same idea. Something that is isoosmotic is some solution that has the same osmolarity as another solution that it's being compared to. So like in an isotonic situation, you don't observe any net movement of fluid. That's equal amount going in, equal amount going out. Hyperosmotic and hypoosmotic then refer to kind of the opposites of each other. Something that has a higher osmolarity than another solution is hyperosmotic, and one that is hypoosmotic is a solution that has a lower osmolarity compared to another solution. Again, very similar to the same ideas that we talked about in hypertonic and hypotonic conditions. Right? In a hypertonic solution, water wants to go towards the hypertonic or the hyperosmotic one to balance out its gradient, whereas in an hypotonic case, water wants to move the opposite direction. We're going to also remember another term, conformers versus our regulators. Some things change with their environment, other things try to stay the same. An osmoconformer is one that tries to maintain an isoosmotic balance with its surroundings. This is very commonly seen in a lot of marine animals. If you live in a pretty salty environment, it is energetically pretty costly to try to fight against that. So a lot of marine animals just go with the flow of the ocean. They don't tend to then lose or gain water due to their surroundings because they're staying at relatively the same balance. And typically ocean salinity is quite stable, so there isn't really a lot of fluctuation that you're going to see. There's no kind of seasonality to how salty it's going to be. If you were to take a look, though, at like this graph on the right to compare the ideas between an osmoregulator and an osmoconformer, a sea jelly would be a really good example of something that is osmoconformer. As osmolarity changes externally, so does the osmolarity of the sea jelly. If we look at the osmoregulators, then obviously their ability is to change the internal osmolarity no matter what the external environment is doing. Even if it gets really, really salty or not very salty, this elephant is able to maintain a pretty stable internal osmolarity. To do that, it means it has to do some regulation, right? If it's in a really hypoosmotic environment, that animal needs to be able to get rid of the excess of water that is trying to enter, right? Think about what a hypotonic solution would look like, hypoosmotic solution. Water's trying to get into that animal, so it needs to be able to kick it out so it can actually balance it. Conversely, if it was in a hypertonic or hyperosmotic environment, it needs to make sure it is taking in extra water to offset that loss. If you're an osmoregulator that does live in a marine environment, you're probably having to drink a ton of water or get at least a lot of water to make sure that whatever salts in that seawater that are trying to lose water from your body isn't going to be detrimental. On these same ideas of osmoconformers and osmoregulators, we're going to look at how they can tolerate changes to the osmotic conditions of their environment. And so you can either be stenohaline or you can be euryhaline. An animal that is stenohaline can really only tolerate a very narrow change in osmolarity, whereas something that is euryhaline can tolerate very big, broad changes of osmolarity. Something that is really non-tolerable, a really good example of that, tends to be a lot of like freshwater fish. Something like your goldfish tends to not be able to handle a salty environment. You cannot plop that goldfish into your seawater tank and expect it to do super well. Euryhaline animals can include a couple of different groups. The one that I usually like to reference is actually a lot of intertidal invertebrates, the ones that are living right on the coast when there's inundations at high tide, but then it gets washed out in low tide and very exposed to the sun. The sun beats down and dehydrates things. Salt levels go really high when that low tide goes, but then salts can go down when tides come in and kind of give that little bit of a flux. Intertidal organisms just have to handle really extreme environments, so they tend to be really good at tolerating huge changes to their salinity. Other animals that also spend their time in both fresh and salt water tend to be euryhaline. Another great example, pretty relevant to the state of Washington just as general, are salmon. Salmon live in the ocean for periods of their life, but then also travel up rivers to mate. They need to be able to tolerate both the fresh water, low salinity, as well as the high salinity of the ocean, so they must be euryhaline. They must be able to tolerate a lot of broad changes. So with those concepts kind of in our head, let's start talking a little bit more on the mechanisms, or how do they actually kind of deal with some of these challenges a little bit. I'm going to start with marine animals first. Marine animals, like invertebrates, tend to generally be osmoconformers. They're not going to try to fight the challenge. They're generally going to kind of go with the flow again, like I've said before. They typically do not face a lot of challenge to balancing water because they just are going at whatever the surrounding environment is. That doesn't mean they don't have to do anything. They do actually have to expend a decent amount of energy actively transporting a lot of different solutes across their body, across their cells. Some lobsters, for example, do this by using active transport to just maintain the magnesium concentrations at a very low level that is pretty generally different from the surrounding environment. So it's not like they're not doing anything. It's just that they tend to not have to worry about losing water in their bodies. Vertebrates for marine animals tends to be a little bit different. I'm going to break this into two groups. I included a phylogenetic tree here just to help kind of understand like what types of organisms we're talking about right now. The first ones I want to talk about are chondriichthyes or chondriichthyans. This group right over here that I'm circling in blue. This includes your sharks, your rays, your skates, and chimeras as well. These are your cartilaginous fish is what their name means. Chondr means cartilage. Ichthyes means fish. So instead of having a bony skeleton, they have a cartilaginous skeleton. How they help balance and manage their osmoregulation is to actually just kind of utilize really high concentrations of multiple solutes. We'll talk about urea here in a bit as a kind of nitrogenous waste, but they'll end up having high amounts in that to kind of help drive water into their body. Basically, make your kind of surrounding cells a little bit more salty. If it's a little bit saltier, osmotic gradient, water follows the salts, helps it drive it into their body with osmosis. They don't actually ever really drink water despite living in a salt water environment because they can do this. They also will get a lot of water just from eating other animals that have water inside their tissues. So that shark survives pretty nicely just using osmosis and eating enough food that is high enough in water. Your osteichthyes, oste like osteo refers to bones, ichthyes means fish, have a different problem. They are actually generally constantly losing water via osmosis. They don't get to use that same urea to help drive it inwards, but instead they excessively will drink water to offset the loss of water being lost via osmosis. So constantly drink, and then make sure you are eliminating a lot of those excess of salts that you're building up, not just through their kidneys, which we'll talk a lot about kidneys, but they can also do this through their gills. For the sake of it, osteichthyes, if this was our cartilaginous fish going this way, this group onwards is technically your osteichthyes. So most marine animals that are osteichthyes onwards, so if you look over here, you can see where that is, will utilize that strategy. Again, if they are a marine animal. So this is kind of my like little fun bit of taxonomy. Notice mammals are right down here. Mammals are technically bony fish because of how phylogeny works. But we are kind of thinking, generally more people like to think about these two groups a little bit more when we think about osteichthyes as a kind of fish. That's marine animals though, and that's a little bit on taxonomy, so you can wait until you take Bio 221 if you want to learn more about taxonomy. So let's talk about freshwater animals instead. Their big problem is generally the opposite of what the marine animals are dealing with. Marine environments, really osmotic, right? We talked about that 1,000 milliosmoles per liter in seawater. Freshwater, much lower in terms of its salinity. A lot of freshwater is in fact hypoosmotic. That means water is constantly trying to get into their body. And that means they have to constantly get rid of the excess of problem of having too much water. They are essentially constantly peeing, constantly urinating, constantly excreting a bunch of water that keeps forcing its way into their body. They effectively do not need to drink any water because enough of it is passing through. Their bigger problem, though, is then making sure they have the proper amount of salts in their body. So they have to make sure that they keep eating things that are salty enough, or they have to rely on those gills, which are still capillaries, in order to get salts back into their body. So if you look at the figure on the bottom here, it's really just showing you that kind of exact opposite difference between, osmoregulation in a marine fish versus something like a freshwater fish. Freshwater fish have to constantly pee very dilute urine, whereas a marine fish probably is urinating very little amounts of urine, but it tends to be much saltier. Both will still utilize their gills to kind of help them balance this, but still, we see those differences. I want to kind of revisit some of these euryhaline fish a little bit, like the salmon, because I just think it's a fun one to think about, fun one to talk about, because they do go between freshwater and seawater environments. Salmon aren't the only euryhaline fish that exists, but I think it's the most interesting one to talk about, because they do have to undergo a very dramatic way in regulating their osmosis, depending on where they are. If they are in the rivers, they will do like what those freshwater fish do. They produce a lot of really dilute urine and try to take up as much salts as they can to help balance that out. Whereas when they are in the ocean, they have to rely on that acclimatization. They have to do things that will help them not lose all the water to the very salty ocean itself. One of the ways that they actually do this is they increase a hormone production called cortisol. Cortisol is generally a stress hormone. And that increase in cortisol actually will increase the ability to secrete salts to keep water in their body, which is done through very specialized cells. So they have nice adaptive strategies in order to make sure that they aren't getting kind of screwed over when they're in the ocean. We focused a lot so far, though, on animals that are just constantly in water. So let's kind of start transitioning our way out of water, and at least for now, in a little bit of a mix. And then we'll talk about land animals right after this. So what happens when water is temporary? If you've ever been around enough places in the world, you'll see that there are ephemeral or seasonal ponds that have water in them in the spring or in the summer, or maybe it's a winter water pool, but then some other time of the year it's completely dry. Or, soil sometimes get these water films that form on them, but if that soil dries, then if you're an animal that lives on that soil film or in that seasonal pond, you need to be able to still balance out, those two very different environmental statuses. For a typical animal, when that seasonal pond dries up, that would cause extreme dehydration because you don't have any water to get. That proper term for that extreme dehydration is called desiccation, basically the shriveling and drying up of some kind of animal. If you take like a really soft like snail or something and just put it out in the sun and you see it bake and dry up and die, or like you've seen a worm probably do that didn't get back in the soil after a rain and then it got really sunny and dried out, that would be a desiccated animal. Now, while those animals don't have the adaptations to help them when that happens, some animals do have really cool adaptations to prevent them from dying due to desiccation. The very famous example of that is our good little friend, the water bear, which is a tardigrade. Tardigrades, sometimes they're pretty well known for being an extremophile, an animal that can live in like incredibly harsh, tough environments. What they are able to do when the water that they live in dries up is they can actually transition into a dehydrated state. And that dehydrated state can stay dormant for decades. It's essentially basically like a dried up flake of an animal that you just have to add water to and it comes back. It's kind of like all those classic like little children's toys that are like the little pills. You put them in water and then the thing gets to grow. That's basically your nice little tardigrade. That term for being able to live without water is called anhydrobiosis. Biosis to live. Hydro refers to water, and refers to the lack of something. So the lack of water, but still able to live. What's kind of cool is if you're to compare the hydrated versus the dehydrated tardigrade state, in the hydrated, about 85% of their body is water. Compare that to the dehydrated one, it's probably more around like 2-ish%. So they can drastically reduce how much water they have. Obviously, if you did that to us, to humans, we would be super dead. Not able to do that. It's a very rare but cool adaptation nonetheless. I did say we would start getting our way out of the water, so let's think about land animals next. Obviously, if you don't have water in your environment, at least you're not living in it, it's not good to help for the retaining of water if you don't actively live in it. So, how do we prevent the extra loss of water? Well, a couple of different ways. First, get a really nice barrier that prevents the water that's inside the body from being able to escape. That could be something like the waxy exoskeleton of an insect. It could be the hard shell of something like a land snail. Or if we think about humans or other kind of like mammals, we have this nice thick layer of skin. We have this keratinized, stratified squamous epithelia, the outer layer of your skin, the part of the skin that you can look at right now on your body. Multiple thick layers of cells. Keratin is a really good water resistant or water impermeable protein that prevents any of that water from getting out of your body. So even though you don't have water constantly bathing you on the outside, you can at least hold all the water on the inside. Second thing you can do, because life is still harsh, is to make sure you are only active when that water loss isn't going to be as prevalent, or it's not going to be as easy to lose water. So be more active when it is really cool out, or when it's really dark out, or when it's higher in humidity. Times when water is not going to more easily go down its gradient and out of your body. When that really hot sun doesn't beat down on you and make you just sweat a lot. Something like this eye that I got pictured over here. This guy, he's called an eye. These nocturnal animals do really well because they aren't, trying to work outside when it's really hot out. You see this in a lot of nocturnal desert animals as well. That's why not a lot of animals out during the day in the desert. Would be really bad to keeping your water around. Thirdly, if you can't do the two of them, or at least if you're already still doing the two and you still need to make sure you're kind of holding things on, is if you can't drink a lot of water, make sure you can at least store water or store energy in some kind of way. So kind of similar to how like the cactus, when we thought about plants, would like to store water to make sure it's not dealing harshly in the desert environments. A lot of other animals have different ways to store water, or at least some form of something that they can still utilize eventually. I like to make sure I point this out though. Camels, people like to often think of them as a great example of water storage. They don't store water in those humps. Our dromedaries, our camels, store that. Bump is fat. That fat can be used for our next point though. So if you can't store water, and you've already done all the other things, or maybe you are storing as much water as you can, how else can you still get water in your body? This is a fun callback to cellular respiration, and that's take the water that you're getting from cellular respiration. Think about that equation, that generalized idea of cellular respiration. It's oxygen plus some glucose molecule, and your result is some carbon dioxide and water. Use metabolism to generate water from glucose and use that water to stay inside the body. It's kind of, I think, a really fun, goofy way of remembering that, like, oh yeah, one of the most important things that your body does is cellular respiration, and its byproduct is a very important water molecule, so you can use it. You don't just have to breathe out all that water, you can keep some of it. But that takes energy. And remember, osmoregulation, just as an idea, takes a lot of energy. We and other animals spend a good amount of our body's metabolic rate on just trying to keep osmoregulation where it needs to be. These are typically a lot of forms of active transport that we are seeing. Now, not all animals need to spend a ton on it. Something like a fish is actually generally pretty well adapted and is using something like 5% of its metabolic output to maintain osmoregulated balances. But some animals that live in incredibly salty environments, like a brine shrimp, can end up spending about 30% of its total metabolic output just to maintain osmotic gradients, maintain osmotic homeostasis. A lot of variability, but still a lot of energy nonetheless. So with that all in mind, we're thinking about energy, we're thinking about how we're moving things. The next idea I want to talk about is some a little bit more into the cells themselves, the tissues themselves that are dealing with osmoregulation itself. And so I want to talk a little bit about what's called the transport epithelia that we utilize, or other animals utilize, in osmoregulation. Animals tend to have a lot of cells in their body, just like we kind of saw with like circulatory systems. Instead of trying to just have the body cells themselves all just get regulated all the time, that's pretty hard. It's pretty difficult to make sure every single cell is at the proper osmotic level. So instead, why not worry about that fluid that bathes all of those cells, whether it's the hemolymph or the interstitial fluid. Remember, blood doesn't actually directly touch the fluid, so it's the interstitial fluid that you're trying to balance, though, as we will talk very extensively, blood is kind of indirectly important for osmoregulation, and sometimes a little bit more directly important for osmoregulation. See this when we talk about how blood and urine kind of go hand in hand in the next video. But if we are trying to make sure that that interstitial fluid or hemolymph, if we're, an insect or something, is making the right osmoregulative changes at the right homeostasis, that means we have to move things from one place to another. So it's going to be really helpful if we have some sort of tubes to help move those things around. Don't forget, these cells are also creating a lot of metabolic wastes while this is all happening. Those metabolic wastes can cause changes to osmolarity that they themselves need to balance off as well. So you're creating problems with osmolarity, you're trying to fix these problems with osmolarity. It is really useful to have a specialized transport epithelia to do all of this. Typically, this transport epithelia is just some complex tubular network to help drain fluids and get them either excreted out of the body directly to that external environment, or at least to some other tubes somewhere else that are going to drain it somewhere else. Get bad wastes away from cells into something else so they can be replenished, and so those wastes can stop being a problem in your body. We'll talk a lot about this in the next video with the kidneys, but to kind of look at it in something like an albatross, like we see over here, albatross and some other marine birds and some other marine reptiles as well have things like these salt glands or salt secretive glands kind of right by the beaks that you'll see. You can also see the zoom in on that nasal salt gland, and then the even bigger zoom in that we see right below it. Something like an albatross is going to be drinking seawater. Seawater, I just mentioned, has a pretty high osmolarity, right? That 1,000 milliosmoles per liter. Milliosmoles per liter. You need to get rid of that extra salt. That's going to be higher than whatever the blood in the albatross is. So as that salty, ingestion is going through the blood, we are going to see the removal of those salts through these transport tubules, these transport epithelia that lets the salts get out of the blood into that tubule, and then that tubule can take it somewhere else, ultimately out of the body through some duct that's connected to the external environment. So if you see something like a picture of a marine iguana or a lot of seabirds, you will see sometimes a really kind of salty white patch right by kind of like the border between their beak and their feathers in their head, because that's usually where that nasal gland is. That is them getting rid of those extra salts. Now, in something like an albatross, these nasal glands, really effective, super good. The reason they can still drink seawater and gain a net amount of water is because these nasal glands are highly efficient. As a human, please don't do this. If you've ever accidentally swallowed seawater, you would know you do not have the ability to excrete that much salt. You would be dehydrated. You would not have a good time. So that's how we do a lot of the moving. We're going to talk about some of these different tubule pathways to get a lot of these metabolic wastes removed in some way, shape, or form. But before we talk about the specifics, we should talk about the different kind of wastes that we do need to get rid of. Typically, these wastes are going to be different forms of nitrogenous wastes. Why is it nitrogenous? Well, because nitrogenous wastes tend to be the extra stuff that's broken down due to metabolism. We'll see this here in a second. We're also going to see different kinds of nitrogenous wastes, and those different kinds of nitrogenous wastes are going to be informed of due to phylogeny, due to your ancestors, how you've evolved, and the kind of habitat that you live in, which will promote certain strategies for working better than others. Think about, again, the difference between a water-dwelling animal versus a land-dwelling animal, etc. So to dive into that, first form of nitrogenous waste we're going to start thinking about is ammonia. Pretty much every animal will accumulate ammonia. It's a very toxic product of metabolism due to the breakdown of all those different biomolecules that has nitrogen. Think back to your different types of biomolecules, what they're made out of, and you hopefully remember that proteins and to an extent nucleic acids have a lot of nitrogen in them, right? The nitrogenous bases versus all those nitrogen groups and like the amine part of a protein. Proteins get broken down a lot. Nucleic acids also get broken down too. That is bad. When that happens, yes, you can reuse a lot of those broken down amino acids and resynthesize new proteins, but when the amino acids themselves break down, you need to get rid of that harmful ammonia. For the sake of it, this is ammonia, NH3. Even though all the animals are secreting ammonia, you're not going to necessarily get rid of it as ammonia. Different animals will convert it into different things. Whether that's urea, uric acid, or as we're going to talk about first, just keeping it as ammonia and getting rid of it. Again, you may remember and at least know ammonia from its effect as a cleaning solution. Ammonia is a very effective cleaner. If you've ever tried to use it for things, it's also very dangerous to ingest. Please don't ever ingest ammonia. It is very, very bad. But if you've used it to clean things, hopefully you can realize that having a buildup of that in your body would not be very good. So, why in particular is it really bad? Well, to do that and to answer that, we're going to talk about the first group that does actually just secrete it as ammonia, just for the first sake of it. The problem with ammonia is often its ion, ammonium, NH4+. NH4+, so basically you're adding another hydrogen there. An extra hydrogen's carrying a positive charge overall for that molecule. That disrupts the oxidative phosphorylation pathway. If you remember, OXPHOS was a part of cellular respiration. If you stop cellular respiration, that's not very good. You don't get energy. If you don't get energy, you don't live. Very bad. And so it is so toxic that most animals need to get rid of it pretty quickly and need to excrete it in a way that it is very dilute. You do not want it getting concentrated in your body whatsoever. So you need to get rid of it a lot. If you need to get rid of it with a lot of water, think about the animals we just talked about that urinate very dilute water. They're going to be the ones that can just get rid of it as ammonia without doing anything else to it. Basically, what I'm saying here is you need to be an aquatic animal to get rid of ammonia, because you need to just be surrounded by a lot of water. You don't have the luxury of being able to consume extra water to help dilute it. You just need that water already there. The nice thing about ammonia is it is pretty hydrophilic, so it does just readily diffuse out of the body. So some inverts, instead of having to have like really complex tubule systems, they can actually just let ammonia get diffused right across their entire body. No problemo. What happens though if you're not an aquatic animal that doesn't have a plenty amount of water all around them to be able to dilute a lot of that urine? You instead are going to have to look at one of our other two options over here. If you're something like a lot of terrestrial animals, something like your nice land mammals, humans totally included, though it does also include some of our more saltwater fish, because remember, aquatic is going to be freshwater versus your marine animals. Those marine animals, mammals as well, ones that don't have a lot of just free water to just, constantly pee with, might have to convert that ammonia into something like urea. Urea, you can see the chemical formula shown kind of twice here. You have two of these amino groups, but it's attached instead kind of to a carbon dioxide, or at least it starts as a carbon dioxide. A little bit of conversions happen to kind of change some of that a little bit. But what it is really, urea, is just a product of ammonia, two ammonia molecules combining with carbon dioxide, and that typically happens in the liver of whichever animal is actually doing it. And then that urea eventually goes to the kidney and gets filtered. Again, this is happening in marine animals and terrestrial animals where they do not have a lot of low solute water. Low solute water would be fresh water if you want to think of it that way. They don't have it, so they can't just pee a lot. The really nice thing about urea is comparatively it is much less toxic than ammonia is, so you can concentrate it more and get rid of it in those higher concentrations, which means you get to pee less. Which is really good because that way you get to hold on to more water. You don't have to worry about, again, constantly peeing. Not every animal, though, is something that is just on the land or just in the water. So let's again think about some of those animals that spend time both in beneficially aquatic environments, but also terrestrial environments, like a good old frog, some good other amphibian. When it is in kind of its tadpole stage or other juvenile stages, when they're fully aquatic, completely in the water, they will actually excrete their wastes as ammonia. But as they start to metamorph into the adult frogs, spend more time on the land, yes, they do still live in the water at times, they will generally switch over to excreting it as urea. There are some nice, cool cascading effects that cause them to be able to change to do that, but they're able to adapt to the environment. Their environment influences their nitrogenous wastes, like I said before. We still have another group if you are looking at that lower right image. These ones instead are going to be secreting what's called uric acid. You tend to find the secretion of uric acid in birds, other types of reptiles, some insects, and then land snails can do it as well. Uric acid is a little bit more complex of a structure, so I'm not going to break down how exactly it all forms, but if you would like to take a look at what that chemical formula typically looks like, there you go. Unlike urea as well as ammonia, it is not very hydrophilic. It does not like to mix with water very easily. So instead, when it's typically secreted, it more comes out as kind of a paste. If you've ever seen a bunch of birds, standing on a pile of rocks and that underneath area where they're all standing is like just chock full of just white, like defecated matter, that is all of that uric acid. Oftentimes that excrement is called guano. Very high concentration of paste like uric acid. Nice thing about it, though, just like how urea is less toxic than ammonia, uric acid is even less toxic. However, urea takes energy to convert from ammonia, uric acid takes even more energy to convert it. So we kind of have this nice little gradient of very hydrophilic, so easy to dispose of in water, but you need a lot of water to do it, versus less toxic, so you can excrete it with less water, and then causes a little bit more energy to be needed to do that, and then you need a lot of energy to convert it to uric acid, but as a result, it is much less toxic, but also doesn't mix with water, so you can retain more water and just kind of defecate this out, or excrete it out, I should say, as a kind of semi-solidy paste. I do like to also point out that even though, yes, we typically talk about uric acid as being, birds and reptiles, or what I just said, all these guys right here, other animals do still make uric acid in some amount. Humans famously have made uric acid because uric acid crystals that form in your body due to the result of metabolism can cause conditions like gout. So still exists, still gets formed. We see this in other animals with some of these other compounds as well. It's not like it's only being made by the groups that I talked about. It's just what's the primary way in which you excrete some kind of nitrogenous waste that was the result of metabolic processes in your body. Kind of wrapping up that point is the whole evolution informs your waste section. Depending on the kind of environment you live in, the different stressors that environment forces upon you, the different kinds of adaptations will tend to solidify in your population as natural selection kind of refines those traits. Obviously, water availability is probably one of the bigger stressors you have to deal with to balance your salt balances, because water is the one that does that for you. If you're something, like a turtle population, compare the ones that live on land, your terrestrial turtles, versus the aquatic ones. Even though they're both turtles, the different environments determine what kind of wastes. Terrestrial turtles tend to release that as uric acid, highly concentrated because they don't have a lot of access to water, whereas aquatic turtles, which live in watery environments, can secrete it as ammonia, because they're just surrounded by water, but then they can also secrete it as urea because sometimes, they don't have as much access, especially the ones that are kind of going in and out of aquatic environments. But obviously, the types of wastes they produce are matching that environmental background. One of actually the best ways to see this is as you can see it in the development or in the environment of the organism as it develops. Because that's kind of the first environment that an organism experiences, right? For you, the first time you were experiencing an environment was actually when you were, a zygote that was then eventually going to get implanted within a uterus, have a placenta as you were starting to develop into a fetus before you were a nice fully fledged person out in the real world. You were in that environment. If you're an amphibian or another reptile or bird that lays in eggs, the conditions of those eggs is the first environment that thing is exposed to. Amphibians, if you ever looked at their eggs, they don't have very hard shells. Here's some frog eggs for context. Therefore, they can release all their wastes as ammonia or urea because those wastes can easily escape the egg. There's no worry, no problem with toxic buildup of those things. If you were a reptile or a bird that has a very nice hard shell, however, that hard shell is not going to let ammonia just kind of pass through. So you're going to need to convert it into something very non-toxic because it will just get built up inside. So it's good to not build up a toxic thing. So you convert it to something like uric acid, which is solid, so it doesn't get to mix in with any of the rest of your body because it's not going to get soluble into the water, and it's already pretty non-problematic. Something like a mammal can get away with using urea because they have a placenta. That placenta is connected to arteries and veins. The wastes that are getting built up as the fetus or embryo is developing can just get carried out by the parental bloodstream and then excreted by the parent itself. Basically, the pregnant individual does the peeing for the fetus. All these different environments are what we see as influencing all these traits in the end. Remember, natural selection are traits being exposed to environmental stressors, and the ones that work better tend to evolve, or I should say, tend to stick around in a population over time, because higher reproductive success is kind of a self-selecting phenomena. So that's a bit about wastes. The next thing that we can get in and think about is the actual excretory system, or at least the urinary system of this. Kind of a little bit more focused. We already talked about excretion a little bit in digestion, but you build up wastes, you get rid of those wastes through an organ system. If we're excreting things, we can call that an excretory system. We talked about those different kinds of transport epithelia. Tends to be different tubes. So generally, no matter what system you're looking at, it's going to be a variation on some tubular theme. Tubes connect to tubes that get wastes from the body to another place, and then outside to the external environment away from the cells. To do all of that, though, we generally can observe some few basic steps. The first thing that needs to happen is some kind of filtration. Your body fluid, whichever one it is, whether it's hemolymph, whether it's the interstitial fluid that kind of interacts with blood in some context, probably needs to meet up with that transport epithelium that will get those wastes to some other place that will get it out of the body. In that filtration step, things like cells, large proteins, or other large molecules won't get put into the rest or into the transport epithelia, but those smaller waste molecules can. Water typically also will cross that membrane. It will form a fluid that we typically call filtrate. We will talk a lot about this in the next section in this unit, or in this chapter. If there's some filtering, we're losing things like water, even though we really want that water to stay. We're actually going to do some recovery of a lot of useful molecules. We basically over-filter our fluids, do more filtering, and then reabsorb a lot of those filtered things right back into the body. Not only is it going to be water, but it's going to be a lot of other really beneficial solutes, things like salts, other nutrients, vitamins, stuff that initially got put out and filtered, go right back into the blood so it can go right back and do something useful for us somewhere else. However, not everything bad gets filtered in that first filtration step, or at least not everything that's excessive that needs to go gets filtered. So you're going to see secondary loss of stuff from the blood in secretion. Secretion is taking other things in the blood, not during the filtration step, and putting it back into that transport epithelia. Sometimes this is done passively. Occasionally you will also see this in active transport. Again, we are going to explore this pretty in depth here in the next kind of video. And then lastly, that fluid, that filtrate that has formed eventually is going to be removed from the body. It's going to be removed from the body typically as urine. Those are the big steps of how you take bad stuff in the body and get it out. So let's look at what those wastes are again, just so we can kind of refresh ourselves. We know it's things like urea, ammonia, could be uric acid as well. Some byproduct of amino acid protein breakdown or nucleic acids, typically due to metabolism. If we're thinking about things like other mammals like us, I like to think about two things like creatinine. Creatinine is a byproduct of muscle physiology. I did say uric acid already, so you already know that that one's there. We also see the breakdown of hemoglobin occasionally, and quite frankly, decent enough. Your red blood cells constantly are recycling themselves. Hemoglobin breaks down. Things like urobilin get formed as a result of that. You maybe consume other environmental toxins if you're drinking alcohols or other things that are stimulants that need to go out of your body. those will be kind of wasted out. And then also extra electrolytes. Maybe if you've had too much salts in your body, you need to get rid of those extra salts, or if you've been drinking just too much water, you might also technically have to get those gone as well. Too much water in this case is technically a waste product. And so there's a bunch of different ways in which these excretory systems will get rid of those wastes. So let's talk about some various excretory systems. The kind of earliest and easiest one to think about are called protonephridia. Nephra is a term you'll see associated with kind of excretory systems. So you're going to see that kind of pop up a few times here. Protonephridia are found in organisms like flatworms that don't have an actual true body cavity like we do, or like a lot of other animals do for that matter. So instead, they rely on these kind of network of dead-end tubules that just open themselves right up to the environment itself. In each of these different branches on these tubules, you'll see structures that are called flame bulbs, because they literally look like the old school like lamp bulbs that have a little flame to them that would help light, places up back in the day. On these flame bulbs, you find two different cells, a cap cell and a tubule cell. On that cap cell are going to be a bunch of different little cilia beating around, little extensions, little organelles coming off of those cells that are helping to draw in fluids from the actual body of that flatworm into the actual tubule cell, which then goes into the tube itself. Basically, you're just directly taking all the stuff in the interstitial fluid and just pumping it through all these different little flame bulbs. In doing that, then you can take that water and whatever other solutes were in there, what other waste products that we just talked about, and form that actual filtrate itself. And then that filtrate just gets to travel through these tubules out to these little openings directly on the body wall itself. So they just have a bunch of these little tiny protonephridia that are all just taking all their fluids and passing them through these tubules and getting them out of the body, kind of nice and connected, just right there. Not super complex. A little bit more developed are the metanephridia of something like an annelid, which is an earthworm. Worm, again, is a term that is not really a single thing. There's a lot of different things that are worms, but for the sake of this, just imagine an earthworm as a good example of an organism that has a metanephridia. A lot of these annelids are segmented, right? If you looked at a worm, you'll see they have like the little bump, all these different segments. And typically there's a pair of metanephridia per segment. They do have a body cavity here, and in that body cavity, there's going to be a bunch of fluid bathing all the different parts of them. And those metanephridia that are connected with these guys are going to be also surrounded by a capillary network, as you can kind of see from this figure on the right over here. Similar to the protonephridia, the metanephridia have these ciliated funnels as well. You see one right over here that is helping to beat around the fluid, which is going to get it to go into that collecting tubule that we see kind of flow in through here, going our way up, and then eventually it's going to kind of loop its way down and around. That is actually going to open up into a larger bladder. You can see this bladder here. So as that fluid kind of comes in and around, gets stored in this little bladder space, and then from there it can leave out through its own little external opening. And you can see though, even though that internal opening passes from one segment to the next segment has its own little funnel here that is then going to lead into the next one and do all that same stuff before leaving again as well. Typically, something like an annelid, I mentioned briefly about how you sometimes see them as super desiccated because they weren't able to get back underground when the watery rain all dried up. That's the non-typical case. Oftentimes those worms are just living in a very water-rich or hypoosmotic soil. So they tend to get a lot of water in their body, or at least much more water goes in than water actually goes out. So they tend to produce a lot of dilute urine because they just have a lot of excess water, so they need to get rid of that excess of water. And what helps, though, is to make sure that a lot of salts are getting lost during that extra dilution of and removal of water is those capillary beds that are right next door. Since they're capillary beds, and we talked about capillary beds, filtration, reabsorption, things go out, things can come back in, they can actually get a lot of those salts to go back into the capillaries from those tubules. And so they get to retain them, use them for whatever else they need to, whereas the water can still just get excreted out along with a lot of those other nitrogenous wastes. That is fairly different than from our protonephridia, because our protonephridia basically are just getting rid of things. They're not really trying to balance any of the salt levels there. They're just create a waste, get rid of the waste. Here we can see the creation of something to excrete, but we also see the reclamation of salts. So we get to see some of that balance of osmolarity in the actual osmoregulatory form. So that's why they're a little bit more fun, a little bit more complicated than our protonephridia. Jumping up one more step, we have the Malpighian tubules. You find these in a lot of terrestrial arthropods, which include a lot of your insects. These guys are just dead-end tubules that connect directly from the hemolymph to the digestive tract itself. So instead of having external environments just kind of dotted throughout their body, here the exit tubule is basically connected to the digestive tract. So you get to kind of combine your wastes all into one area. So we've talked about hemolymph before, bathing the cells, bathing the body of something like an insect instead of like the kind of interstitial fluids that we've seen a little bit in our other animals. Here, those Malpighian tubules just are connected to this different hemolymph areas. Hemolymph can get filtered into them. Salts, nitrogenous wastes, all that same jazz that we've talked about before, water included, gets to go into this filtrate. As it goes into the filtrate, it gets to enter the digestive tract. We've talked about the digestive tract. It has the ability to reabsorb things, good helpful nutrients. So here's where we get to see kind of that reabsorption step instead of seeing it happen just directly within the Malpighian tubules. And since it gets to go into the digestive tract, it actually ends up being really good at reclaiming a lot of that water, because as you take a solute, pretend it's something like sodium, you reabsorb it into the hemolymph. Water wants to follow that gradient as well. Again, water goes from an area of high to low concentrations relative to itself. So if you take solutes away from the water, you've now kind of artificially bumped up the concentration of water, so it gets to go down its own gradient. Those wastes that do remain in the digestive tract tend to be mostly insoluble, tends to be uric acid combined with the other feces from the actual digestive tract itself. And what I should kind of clarify actually is, I said filtration in a little ways. It's not really the same kind of filtration that we do see when we're going to talk about our next group in mammals. It actually is really just a form of secretion, which is kind of unique. Instead of relying on that filtration step, we just secrete things into those tubules and then let the digestive tract take over from there. Where we get into something a little bit more familiar for us are the kidneys, which you see in a lot of vertebrates and a couple of other chordates as well. We're going to, again, contextualize it in the vertebrate we probably all know at least a little bit more than the others, which is ourselves, humans. Kidneys are really highly organized system of tubules, basically tubules connected to other tubules that often get associated with things like capillaries and other ducts as well. They themselves aren't just connected directly to the external opening, they instead will lead to other organs that can transport larger amounts of waste fluids all at once. Things like a ureter, like we see here, which will connect to something like a bladder, which can store a bunch of urine until it's a good, safe time to release it, and then a urethra, which actually expels the wastes itself. If we take a look at that second image here of just the generalized structure of the kidney, and we'll talk about it a little bit more, you can see how blood enters the kidney. If we take a little zoom in on that as well, we'll see how some of these smaller arteries and veins are connected to some capillaries to these structures here. These structures in yellow are going to be called nephrons that are going to do all of those different filtrate, secrete, reabsorb steps that we were looking at. Eventually produces urine that is going to collect in a larger structure here that then gets to go out through that ureter to transport it to the bladder to then get out. And we'll look at all of these different pieces here as we move on. So, to give you that kind of just overall structure of a kidney, for most organisms you have paired kidneys, a left and a right, though there is variation within that. So I won't always say that's the only possibility, but for something like a human, you have typically two kidneys. Your kidney has two different tissue layers. You have the outer renal cortex, and you have the inner renal medulla. The renal medulla is organized in these kind of triangular pyramids that we see me circling here, whereas the cortex kind of surrounds it on the outer border, but then you do see these renal columns of cortical tissue that kind of just dives inwards a little bit there. Going into the kidney, we have the renal artery. Going back away from the kidney, we have the renal vein. The renal artery is taking blood that will get filtered, and the renal vein is returning blood that has been filtered. The urine itself that is being produced is going to get stored in this space right here, known as the renal pelvis. What really happens is urine is getting formed right at the base, eventually through these structures called the nephrons, and as it's coming in, it's going to connect to these tubule structures here called the calyces, the minor and major calyx. Calyx, I'll also definitely restate that in the next video, but it connects all these two together to eventually lead into that renal pyramid, which will, or sorry, not renal pyramid, into that renal pelvis, which will drain into the ureter. But we're not actually talking about how that urine was getting formed, though, or how that filtrate was getting formed in the first place. So we need to take a zoom in now, kind of towards the border of the cortex and the medulla, to see and describe what a nephron is. Nephrons essentially are the functional unit of your kidney. You typically have about 1 million nephrons in your body. We see two different types exist. These types are the cortical and juxtamedullary. Cortical and juxtamedullary differ mostly in their kind of structure where you find them. As their name sort of implies, the cortical nephrons are ones that are mostly contained within the cortex, and then a little bit of them extends into the medulla, which you see right here, coming kind of back though, mostly in the cortex. They tend to be much shorter in length. Notice this kind of downwards looping part in particular. We'll talk about what that is here in a second. But their job is to actually produce a majority of the urine that an animal will typically have. The other type that we see, I should say of a kidney-containing animal, just for clarity. But though, as I was saying, the juxtamedullary, our other type here, are more distinguished by a longer kind of loop that we see here that extends much further down into the medulla of your kidney. Excuse me. You can also see that in this top figure a little bit. I don't think as clearly, but I wanted to still reuse the same image so you had that as context. Cortical versus deeper juxtamedullary. Juxta, for the sake of it, means like to be by something or around. So like it is a round or by the medulla of the kidney. These ones tend to help a lot more with the concentration of the medulla. We'll talk about osmolarity in the medulla again in the second video on this one. We'll spend a decent amount of time with it. We'll talk specifically about pathway through a juxtamedullary nephron, and it will then also help with the actual water conservation process, making sure that if you are dehydrated, you are getting most of that water back. If we're to take a look at the far right image on this one, we'll see this one in the next slide as well. We can see kind of the basic layout of it. We have some artery that's going to be taking blood into the nephron. Then that artery leaves and turns into a capillary bed. That capillary bed is going to wrap around the different parts of this nephron here, and that's where we're going to see things like the reabsorption and secretion of a bunch of different fluids. It's at this kind of bulby spot right here that we're going to see that filtration step happen. So, to get into these different parts next, we're going to actually just look at each part of the nephron kind of one by one here. So to start, I'm going to break it off into two parts. There's going to be what's called the renal corpuscle, and the second part is going to be the actual tubule component itself that I'm going to call the renal tubule. There's that kind of renal prefix again. For the sake of it, we've seen this now a few times, just refers to things of the kidney. The renal corpuscle is made out of two components. This is what I've kind of broken apart right over here. We have the glomerulus, this kind of thing right here. This glomerulus is actually just a capillary bed, kind of intersected from this artery that's coming in. So artery comes in, turns into a capillary bed, and then it leaves still technically as an artery. But what we know about capillaries is we know things can move in and out of them, so that is going to be the site of filtration. Specifically, the filtrate that gets formed due to the filtration is going to collect in the other part of our renal corpuscle, known as our Bowman's capsule. The Bowman's capsule is this structure that is surrounding the glomerulus. Here it's been cut in half. On the image on the right, you can see how that Bowman's capsule surrounds the glomerulus on the inside. After that filtrate is formed, it is going to leave the renal corpuscle and go through the renal tubule. The renal tubule is made out of a few different parts. The proximal convoluted tubule, sometimes also just called the proximal tubule. Then it's going to go from there down and then back up what's known as the loop of Henle, or just the nephron loop. The part that goes down is the descending limb. The part that goes back up is the ascending limb. For the sake of it, proximal convoluted tubule, you can call it the proximal convoluted tubule. You can abbreviate it as the PCT, or you can call it the proximal tubule, or just PT. Those are all totally fine abbreviations. Those are all common enough vernacular that people know what you're talking about here. Convoluted, and again, I'll probably restate in this next one because you guys might watch this on a different day, so it's always good to repeat and repeat. Convoluted here is because it is convoluted. It is going back and forth. It is not just a straight tube. It is zigzagging back and forth. You can kind of tell again as I retrace this that it is not a nice clean straight line. The loop of Henle though goes down, comes back up. If there's a proximal convoluted tubule, for the sake of it, proximal just means something is close. It is closer to something. It is closer to the renal corpuscle. And then distal, in our distal convoluted tubule, means further away or further from. So it is the further of our squiggly lines. We have the proximal convoluted tubule that leads into a nephron loop, and then the distal or the further tubule. Same thing like the proximal tubule. You can call it just the distal tubule, or just DT, totally A-okay. That distal tubule, though, drains into a structure known as the collecting duct, sometimes also just abbreviated as CD. When it comes to the collecting duct, it shows it here. Again, I will probably just repeat all this again for the second time we do this in the next video because repetition is key here. It is not just one nephron per collecting duct. Multiple different nephrons will lead into a single collecting duct. You can see that in some of the other images here. Here is again one collecting duct. There's multiple different nephrons that are leading into it. You can even see that here in just this example. Here is two of them leading right into that same collecting duct. That's the tubule part itself. That is the actual kind of, nephron itself. That is where the filtrate is actually going. The filtrate will travel down the collecting duct. At some point within the collecting duct, it won't be able to be altered anymore. You won't be able to change the concentration, the osmolarity, the amount of water, salts, yada, yada. After it gets that point of no return, that's when you will start calling it urine. But you need some fluid to actually change. You need some fluid to osmoregulate. I previously said, it's the interstitial fluid that we're worrying about maintaining the osmoregularity because the blood doesn't really directly come in contact with it. But to actually do that, we osmoregulate the blood. And so blood has to come in to the nephron and then go away from the nephron. As blood is going in from a renal artery, it's going to turn into what's called eventually the afferent arteriole. The afferent arteriole enters, becomes the glomerulus, filtration happens, and then it leaves as the efferent arteriole. Efferent for exits the glomerulus, or exits the renal corpuscle. Afferent goes into it. After the efferent arteriole leaves, it turns into a capillary bed, right? Arteries turn into capillaries. The capillaries that are near the proximal and distal tubules are called the peritubular capillaries. Peri, like perimeter, means to be around something. It is around the tubes of the nephron. However, those peritubular capillaries will dive down alongside that nephron loop as it goes down through the medulla and then back up. And the vessels that we find there are called the vasa recta. Those vasa recta are the end of the capillaries, so they'll return to a venule. That venule will then return into a vein, the renal vein, and then that renal vein goes back into the rest of your blood circulation after having already been filtered. We'll talk about what the vasa recta, what the peritubular capillaries are all doing, how we are actually doing all this filtration, secretion, reabsorption in the next video, since this one's already an hour long. But I want to leave on one little point here. Kind of like we've already talked about, different environments are going to give you different kidney structures for those animals that actually have kidneys. Birds and mammals have juxtamedullary nephrons. But mammals tend to have theirs dive deeper into the medulla. Mammals are good at concentrating their urine to really good amounts. Birds don't have the ability to concentrate that urine in the exact same way, though they can conserve it differently via the formation of uric acid instead. So different adaptive strategies still get to a good end point, just different ways that you go about it. Other reptiles will sometimes only have cortical nephrons that are generally isoosmotic or even hypoosmotic to the urine that they're trying to produce, but they can actually reabsorb water across different epithelial surfaces, like their cloaca, which can then again, prevent them from having to lose water in other places. The fact that they are not making necessarily a super concentrated urine isn't a problem because they can also just get water other ways and get rid of water other ways. A lot of freshwater animals, which would include some fish and other amphibians, we already talked about how they're producing a ton of dilute urine. So they need the types of nephrons that produce urine. So they're going to have just a bunch of those cortical ones. They're not going to necessarily need as much as those juxtamedullary, which helps establish salinities and helps concentrate things. What we'll see here in the next video is that the distal tubule is going to be really important for reclaiming, getting a lot of those salts back into your blood flow. So they're going to really utilize those distal tubules because they need to get a lot of salts back, because they do just have to constantly pee because they're just, inundated with their own water. Marine fish obviously are a little bit different. They need to not urinate nearly all that much because they are just surrounded by salts. So they're going to have a lot less nephrons in general. They don't need to form as much urine, and they're not going to necessarily rely on having something like a distal tubule or even a glomerulus, which would filter their blood, which would be problematic because they don't want to get rid of all the water they do have. They want to produce super little amount of it. So that variation is critically important for them. As I get into this next video though, we are going to use a human kidney as a reference. So please just understand what I talk about is in our kind of reference point and not the fact that obviously animal variation exists, different kinds of animals, different stressors. We've talked about that kind of ad nauseum here. But that finally gets us to the end of this video. Next video, I said, is going to be the stepwise process of what actually happens inside a nephron, giving you a little bit of a refresher along the way, because I know this chapter does end up tripping up people in a lot of ways. It's a lot of this thing moves in, then this thing moves out. So ask a lot of questions, but good luck in the next video. See you later, as always.